If you monitor the membrane potential of a neuron in the brain by putting the tip of an electrode in or very near the neuron's membrane, what you'll see is that the neuron isn't firing action potentials at a constant pace, or just at random. Instead, the firing rate of the neuron is related to something in the outside world, or to some behavior of the animal. The firing rate of the neuron is encoding and conveying information, either about the sensory world or some internal state or process in the brain. You can count up the number of action potentials a neuron experiences in a fixed amount of time, divided by the length of that time window, and that number is called the firing rate of the neuron. This will be measured in action potentials per second, or hertz. The idea that the firing rate of a neuron changes based on some external variable is called rate coding, because the firing rate of the neuron encodes information about something else. Often, you'll look at when the neuron is firing action potentials relative to some outside event that you as an experimenter can control. I'll show you an example where the experimenter is recording the activity of a neuron, another term for measuring the membrane voltage, while presenting specific visual stimuli to an animal. Here we have something called a raster plot. Each vertical line represents an action potential fired by the neuron, and the x-axis represents time. Usually you'll take multiple repetitions of some single event, for example, the presentation of a visual stimulus, and line up the raster plot showing the neuron's firing rate over time relative to the onset of that stimulus. Each repetition of the same event would be referred to as a single trial. So in this case, you can see that the neuron has a low baseline firing rate when there's nothing on the screen, but shortly after the visual stimulus comes on, the neuron will fire more spikes. So this neuron fires in response to a visual stimulus of an vertically oriented bar. Another thing you might notice is that the exact total firing rate and the exact times at which each action potential occurs varies from trial to trial. The responses of neurons are generally not perfectly repeatable, but instead are slightly noisy, meaning variable, due to random variability in the many biological processes contributing to that particular neuron firing an action potential. Another way to look at this same thing would be, instead of showing each action potential in each individual trial, to show the average firing rate of the neuron over time across multiple presentations of the same stimulus. This is called a PSTH, short for peristimulus time histogram. Now we can test the effect of a bar with a different orientation, so horizontal instead of vertical, and examine the firing rate of the neuron again. In this case, you can see that the neuron still responds to the horizontally oriented bar, but not as strongly as it did to the vertically oriented bar. So we would say that the neuron is selective for, or tuned for, or prefers the vertically oriented bar. There are lots of different ways to express this idea of what a neuron responds to. You might hear that a neuron prefers a particular stimulus, is selective for that stimulus, is tuned to a particular property, responds to, encodes, reflects, represents, is driven by, or even colloquially personifying the neuron as liking a particular stimulus. The example I showed you was for a neuron early in the visual system, but there are also neurons that encode all kinds of other things. For example, faces, smells, anticipated reward, fear, location in space, etc. So an action potential has a different meaning in different neurons, depending on what that neuron is selected for. If you know something about what that neuron responds to, then an action potential in that neuron can tell you something about the outside world. In sensory systems, activity of that neuron tells you something about a sensory stimulus. For example, in the auditory system, different neurons respond most strongly to sounds of different pitches or frequencies. In the motor system, each spinal neuron projects to a specific muscle and causes it to contract. So activity in a neuron that connects to the biceps will have the opposite effect of a neuron that connects to the tricep. This case, where activity in two different neurons has different meanings or effects, ultimately based on their physical connections with other neurons, or in this case muscles, is an example of a labeled line type neural code.